Functions of major enzymes and other proteins of replication machinery. A number of enzymes and other proteins are required in the replication of DNA. These proteins assemble at the origin of replication. The major enzymes involved in the DNA replication are helicase, topoisomerase, primase, DNA polymerase, DNA ligase. There are several other proteins in the replication machinery, including single-strand binding proteins (SSB). Helicase. These are the enzymes that unwind the double helix and separate the two strands of the DNA molecule with the expense of energy as ATP. The separation occurs by breaking hydrogen bonds between the base pairs of the two strands. This is important for exposing the two strands to function as template for new DNA synthesis replication. Topoisomerase. These enzymes are working ahead of the direction of DNA synthesis. The untwisting of the strands at one place causes further twisting and staying on other places. Topoisomerase enzymes introduce breaks on one or both strands of DNA. Twist the molecule to relieve the strand, then reseal the cut ends. Single-strand binding proteins (SSB). These protein molecules bind to expose single-stranded DNA to prevent repairing of the separated DNA strands and to stabilize them. If the two strands repair, then they cannot serve as template for new DNA synthesis. Primase. In order to synthesize a new DNA strand on a template, complementary deoxyribonucleotides should be added one after other in the correct sequence. This is done by DNA polymerase. But DNA polymerase can add nucleotides only to the 3' of an already formed nucleic acid chain. For this, a small fragment of nucleic acid chain is sufficient. This fragment is called primer. Primase is a type of RNA polymerase which can initiate synthesis of RNA on a DNA template by adding ribonucleotides. Primase adds a short RNA primer on the DNA template, forming DNA-RNA hybrid, facilitate the action of DNA polymerase. DNA polymerase. There are several types of DNA polymerase. One type of DNA polymerase initiates DNA polymerization by adding a deoxyribonucleotides to the 3N of a primer. Continuous polymerization on 5 to 3 direction, elongating the new DNA strand with deoxyribonucleotides with base complementary to template. This DNA polymerase is almost 100% error-free in adding carried complementary nucleotides to growing strand according to the nucleotide sequence of the parent DNA strands. However, one error in 10 to the power 5 added nucleotide is possible, has a proofreading mechanism, therefore can correct its own mistakes. The error rate will be reduced by 100,000 times to 1 in 10 to the power 10. Therefore, the daughter DNA molecules are almost identical to the parent DNA molecule and of course to each other. When a wrong nucleotide is added to the growing chain of DNA by DNA polymerase, this mismatch is identified by the same enzyme. Stop adding the next nucleotide, removes the incorrect nucleotide, exonuclease activity. Continues again its polymerase activity. This is called as proofreading activity of DNA polymerase. Another type of DNA polymerase identifies DNA RNA hybrid, removes ribonucleotides and replaces those with deoxyribonucleotides. So replace the RNA primer with DNA. Now the DNA segment is incomplete, but DNA polymerase unable to correct the ends of Okazaki fragments. Results gaps. DNA ligase. In the DNA synthesis, it joins the newly synthesized adjacent fragments, making a complete strand by the formation of a phosphodiester bond. It seals the gaps of the newly synthesized DNA strand.